Hello everyone, this Doc Ron Bio video is about cell communication. Here I hope to focus on the content of the curriculum for the 2019-2020 school year, so it's the new curriculum on cell communication. Cell communication actually goes by a lot of different names. The College Board calls it cell communication, but a lot of times you'll hear it generically called cell signaling, signal transduction, cellular talking. And in my opinion, all these names are interchangeable, so whatever you are most comfortable with is fine. This video discusses basically how cells relay messages in order to maintain homeostasis. So considering the curriculum, topic 4.1, cell communication, when you dig into the course and exam description, the CED, what I refer to herein as the pink book, it highlights the certain things that cells need to do to communicate. And generally speaking, a cell needs to receive signals, respond to signals, generate signals, and in most cases transmit those signals. I look at this as two parts. So when the signal is being transduced inside a cell, it is received, relayed, and eventually causes some sort of important cellular event, usually gene expression and the production of some sort of protein that does something for the cell. I know that sounds very generic, but it kind of is a generic process until you talk about an actual pathway. So looking here at this figure, essentially on the left, the cell is talking to itself. So when you see on the outside here, receptor interacting with a signaling molecule or ligand, it'll lead to some sort of transduction inside the cell, and eventually the production of some sort of protein. Okay, so the cell is essentially talking to itself. All of this is intracellular. Now for the fourth part, if the cell wants to relay this message to another cell, we go into another separate part. So here I'm showing you basically this endomembrane system here. So there are two different pathways obviously inside the cell to make proteins, those with ribosomes in the cytoplasm and those with ribosomes on the ER. So if we want to send that message to another cell, so you can see here the blue protein is being exocytosed, secreted, in order to talk to this neighboring cell. In order to do that, we need to go through the endomembrane system. Okay, so that fourth part would be the actual transmission of a signal to another cell, and in this case, talking to other cells. So this leads me to a quick aside about AP biology. So it's rare for a question or an FRQ in AP biology to discuss just one aspect of biology on its own. So it's usually talking about multiple ideas. So don't get caught up in just talking about one thing. You want to be able to talk about several things at once. So for example, in this case, we're talking about the endomembrane system and how you have to shift from talking about proteins in the cytoplasm to proteins being secreted. So don't miss a chance, especially when it comes to cell signaling, to talk about multiple biological concepts at once. So here we're talking about the endomembrane system and how proteins are secreted and transported to the outside of the cell. But going back to this figure, the cell is generated and transmitted a signal to its neighboring cell. So in this way, a cell can convey important and helpful information about the environment to other cells. So there are so many ways to do this when it comes to signal transduction in biology. There's no particular signal transduction pathway that you have to memorize or know. Just understand that there's a lot of different ways to do this. And this video will give you some examples from the pink book that you can go over and be familiar with going into the exam. Okay, so there's a lot of videos out there on YouTube that will go over the specifics of some of these examples. This video really isn't for that. It's just to get a general lay of the land and to talk about some of the general concepts when it comes to signal transduction. Okay, so the College Board, remember, is just going to test your knowledge of important general themes. And I know that is a lot still, but you don't have to know a lot of specific biological pathways and reactions, so you don't really need to commit that information to your memory. You really just need to draw upon very important large ideas and concepts, which I'll hopefully highlight here for you. Okay, and I also want to mention before we go on that what I've noticed in the past few years is that cell communication as a biological concept has become more common on recent tests, and I expect this trend to continue for reasons that I'll mention in this video. Cell communication, signal transduction, can basically involve any aspect of life science that you've learned about this year, so it's a really important topic. Okay. So here are some other examples from the book. You're not going to have to know any specific pathways. Really stick with broad examples like these shown here. So 4.1 also goes on to talk about these illustrative examples. One of the ones that I'm a really big fan of is the immune system. I'll tell you why in a minute. But also cell junctions, nervous system, and quorum sensing. Those are some of the really important ones that I'll mention in here. So let's talk about how cells can communicate. Okay, cells can communicate through direct contact. So this example of cell junctions is a really good one, I think. These are examples seen in plants and animals. Here I'm showing you gap junctions inside animals and plasmodesmata inside plant cells. And what these adaptations do is it allows neighboring cells to communicate directly through openings in the cell membrane and cell wall. So basically it allows them to directly share small molecules from their cytoplasm, such as ions, sugars, other solutes, water, and sometimes even proteins and RNA. So in this way, these cells can know a little bit about the neighboring cell environment and act upon it. So if ion levels are low, they can read that just from direct contact through these cell junctions. Another example of direct contact cell communication can be seen in the immune system. So in this figure, you see an antigen-presenting cell, quote, showing T cells what foreign pathogens it has encountered. So this direct contact will help inform the T cells about what is going on inside the cell. So we see here this APC cell is showing the T cell through direct contact what is encountered. And here you're going to see that set in motion a cascade of events inside the T cell that will basically help it go on to interact with other cells in the immune system to neutralize this threat. I know, again, that's pretty vague, but I don't want to go into too much of the immune system here. I do think, my approach for cell signaling, I'm going to highlight some general themes here, and then I'm going to move on to some of the important aspects of cell biology down the road. So you guys will be watching videos on the immune system, on the nervous system over the next couple of days. I think those are both systems that are good examples of cell-to-cell communication. So we'll cover that from the cellular aspect. And I should note here again, too, that I really think this is going to be an important topic, this immune system, due to the fact that here in 2020, we're just learning about what SARS-CoV-2 is as a virus and what its biology is. So I would expect some sort of signal transduction immune system question in these two FRQs that UAP biology students will be taking here shortly. So cells can also communicate over short distances. Here you see two strains of yeast that are sending signaling molecules out with the hope that two different strains, that each other strain can detect the other one. So here the alpha strain sends out alpha factor, and the A strain sends out A factor. The message is simultaneously received on the other yeast strains, and when that happens, the cells move toward each other and fuse to create a more genetically diverse offspring. So yeast mating is an excellent example of cell signaling across a very short space. This is another excellent example of cell communication over short distances. And again, the nervous system is a really good example of signal transduction cellularly. Here you see a neuron, which is the functional unit of the nervous system, secreting neurotransmitters into the synaptic cleft, which can bind to receptors on neighboring cells, thereby passing a message along to those neighboring cells. So again, for my students, we will go into this in a little more greater detail later. So here are some other examples from the Pink Book that you could use to illustrate cell communication in accordance with 4.1. They mention plant immune response, quorum sensing in bacteria, morphogens in embryonic development, and the endocrine system. So I'm not going to go over any development here in this particular video, but keep in mind development involves a lot of cell communication, with the end goal being a developed, fully formed organism. And I don't know many classes that go into a whole lot of development in AP biology, but a really fascinating field. The endocrine system, again, excellent example of cell communication, but don't spend too much time on that because it's not worth your time, and I'll mention why that is in a minute. Okay, so cells can also communicate over long distances. Again, no better example than the endocrine system, which has hormone-producing tissues, sometimes very far away from their target cells and tissues. So the hormones in those cases are secreted into the bloodstream, travel great distances to interact with and affect their target cells. For the most part, the College Board will provide you with the details of whatever endocrine system question if they do ask us that. So again, don't spend too much time on those examples. Okay, so here we're going to look at an introduction to essentially what signal transduction is. So you'll have to know the general makeup of a signal transduction pathway and how it works. The College Board has used this figure before in test questions. It's a very popular figure in textbooks, so I think it's important to understand how it works and what it shows. So when you're looking here at this generic signal transduction pathway, the first part that should stand out is what we call reception. So here, this is a case of a receptor molecule interacting with a signaling molecule or ligand, and that receptor will start in motion the signal transduction pathway once it receives that extracellular message in this case. The second part here is transduction. And transduction is basically a series of molecules, many of which are proteins, that relay messages received from the receptor. And then 3 down here, we have some sort of response. So some sort of cellular response or cellular activity that is the end goal of all of this signal transduction pathway upstream here. Okay, so in a quick note aside, keep in mind that all receptors are not necessarily found on the cell membrane. For some signaling molecules, the target receptors in the cytoplasm. These signaling molecules get through the membrane based on membrane biochemistry. So I'm basically talking about lipid molecules that go directly through the membrane because they can get through the hydrophobic tails and interact with receptors inside the cytoplasm. So again, another interesting biochemical aside, if you will, another way to incorporate other information from the class. So moving right along, I want to talk a little more about some of the other molecules involved in these processes. So for me, the most important idea here when it comes to signal transduction is structure function. That's a theme you really want to focus on when it comes to cell communication, because it enables you to discuss so many other parts of biology. So an important pair of molecules that help activate and inactivate proteins in transduction pathways are kinases and phosphatases. So these are these intermediate molecules that we were talking about in the transduction portion. So if you're following along here, we have basically a kinase phosphorylation cascade. So as soon as that message is received at the membrane, that's going to activate a key relay molecule, which is going to activate this inactive protein kinase. So here it is activated, and what we see is kinases activating kinases. Once that is activated, it'll activate another one, and so on and so forth, until it gets to a terminal protein that can do something like translocate to the nucleus. So oftentimes these proteins at the terminus of the transduction pathway are transcription factors, which go on to mediate some sort of cellular response. Okay, so these pathways involve energy. So you probably noticed it right here. The phosphate that's being added to proteins is taken from an ATP. So kinases add that phosphate, which alters the protein structure. So it takes that inactive kinase, alters its structure, and makes it active. Phosphatases do the opposite. So phosphatases, shown here, here, and here, are enzymes that remove phosphates from molecules. So they essentially undo what kinases do. They remove phosphates. They return the protein's conformation back to its normal conformation, and generally they inactivate proteins. So there's that turning on and turning off of signaling molecules inside the signal transduction pathway. It's a really important note. And then a really quick aside, because 4.2 asks us to talk about some of the molecules involved in signal transduction. There are other molecules that play a signaling role. We call them second messenger molecules. They can be proteins, but really what we're talking about here are things like cyclic AMP and protein kinase A that continue to pass the message along. Calcium ions even serve as important secondary messengers in some pathways. So again, that's not something that you necessarily need to memorize. You take it on a pathway by pathway basis, and most likely the College Board will tell you if these particular molecules are involved in your pathway on the test. So topic 4.3 covers some examples of signal transduction and emphasizes the role of the environment in starting transduction. Okay, so it also talks about some of the cellular responses downstream, but I really want to note an important example of how an environment can start a signal transduction pathway. So I'm going to go here to quorum sensing in bacteria. It's a really good example of signal transduction in certain environments. Okay, so during quorum sensing, bacteria must reach certain levels of density in order to perform an important cellular response. So they are able to sense the density of the cells around them by the density of certain signaling molecules. Okay, so I've noted those signaling molecules here in red. So the left shows when bacteria numbers are too low, and consequently the signaling molecule concentration is too low to cause the cell response. So here we only have 3 signaling molecules, so not enough to start the process. On the right, however, because there's a higher density of bacteria, you're able to reach a quorum in which there are enough bacteria to create a critical concentration of that signaling molecule. And the types of downstream signaling cellular activities that occur because of the signaling molecule can vary depending on the bacteria. So some examples of that include toxin production to coordinate infections, biofilm formation, and even bioluminescence for some bacteria. So when you stop and try to incorporate the evolution of this, it makes sense that bacteria have evolved mechanisms to wait until there are enough cells to be effective for something like producing toxins for infections or for biofilm formation or bioluminescence. Okay, so the second part of 4.3 discusses examples of cellular response. So it's often a safe bet that the downstream cellular response will be gene expression in many signal transduction pathways. So generically, if you look at these pathways, they often end in a transcription factor that translocates to the nucleus. I mentioned this before. So here you have a transcription factor. And what it does, it's going to move from the cytoplasm into the nucleus where your genetic material is located. Once it reaches the nucleus, it can alter transcription of those genes in your DNA. So for example here, if this transcription factor goes to the nucleus and activates expression of a proto-oncogene, well, in this case, activation of this signal transduction pathway would result in an increase in cell division. Okay, so that's your downstream cellular activity that we're pointing to. Okay, so 4.3, the other examples of downstream cellular responses that they mention, again, are some of those developmental pathways. So SRY expression leading to male development, Hox genes leading to normal development in organisms. Again, developmental pathways play a big role in signaling. The signaling pathways often lead to proper development. And then we talk about ethylene gas and fruit ripening. Another excellent example that we don't really have time to talk about. I'll mention fruit ripening here towards the end when we talk about positive feedback, but let's get back to these slides. So topic 4.4 asks the question, well, if signal transduction pathways work this way, what happens if we change a certain aspect of the signal transduction pathway? Okay, so for example, what if one of the proteins that plays a role in this pathway is in some way altered? Okay, so cutting to the chase here, the quick response is that, well, whatever the pathway is supposed to achieve will not be achieved. But let's take a look at two biological examples and how this plays out. So the first scenario is mutation. So if there's a mutation to your DNA and the protein that's expressed is in some way different, for example, say you mutate your DNA, it changes a certain critical amino acid in the protein, which alters its structure and consequently its function, you're going to not get that pathway working correctly. Okay, so again, I really emphasize this idea of structure function. There's really, it's a very important concept in biology. So let's go ahead and say that this protein is in some way, this receptor protein is in some way mutated here where it should bind the signaling molecule. So if the receptor protein in a critical spot, in the spot that's supposed to bind this signaling molecule, this ligand, if that doesn't do that, you might not get binding of this signaling molecule. If that doesn't happen, you don't get transmission of that signal to the next protein in the cascade. So, for example, if downstream we were going to do cell division, well, we've blocked cell division by not getting ligand binding and not transmitting that message to the signal, to the next signaling molecule in the pathway. Another way this can be achieved, the second scenario, if you will, is if these proteins are inhibited. Okay, so this is a really important way to connect biochemistry. And by the way, looking for biochemical inhibitors of signaling molecule pathways has made billionaires in the pharmaceutical industry. So if you're looking for, a goal, a job down the road, understanding how this biology works can be lucrative. So each of these proteins, again, in this pathway is potentially a target for inhibition. But let's stick with this molecule up here again. So if your receptor, which should bind this ligand, instead has a different competitive inhibitor that can get in there and block the actual ligand from binding, it's competed at the binding site. Well, same thing is going to happen here. You're not going to get movement of the signal transduction pathway. It's basically blocked very much upstream. And again, if we're trying to affect cell division, no cell division. Okay, so again, it's another important example of how this signal transduction pathway works. Okay, so again, let's take this home and emphasize it. So if you alter your signal transduction pathway at any step, whatever you are trying to do, cell division, even if it's the very last step, is not going to occur. That can happen at any of these steps here. Okay, so going towards the end here, talking about topic 4.5, where they talk about feedback. For my students, we've talked about feedback before in AP biology, not just in the context of signal transduction, but it's a really important theme and is relevant at this part as well. So there are two types of feedback, positive and negative feedback. We'll break it down here and discuss examples of each, but I should mention here that they start to talk about a different set of information. So they say here, the timing and coordination of biological mechanisms involved in growth, reproduction, and homeostasis depend on organisms responding to environmental cues. Okay, so they're switching their enduring understanding here to ENE3, and that's really important to note. So let's talk about feedback here. So first, positive feedback, with the examples in this case being onset of labor and fruit ripening. So when a woman has a child and when fruit get ripened, follow a similar positive feedback mechanism. Okay, so obviously the molecules that are involved, the hormones that signal this, are completely different, but I just want to talk about this from a homeostasis positive feedback perspective. So if this line represents homeostasis, okay, so if we're looking here at this black line right here, for homeostasis, like we're sort of hovering over this line at any point. However, for things like labor and fruit ripening, there's only one way to go when it comes to these two biological activities, okay? So they're both based on positive feedback. So each of these pushes away from homeostasis. So they're going to involve hormones that push away from homeostasis towards the goal of childbirth and ripe fruit. Okay, so there is no going back. You're not going to return back to homeostasis where you don't have labor occurring or where your fruit was not ripe. Okay, there's no going back. So in other words, positive feedback, any of these mechanisms that do this are going to push towards the end goal. There's no going back. The pathway will push forward until the goal is achieved. And the goal of this, the goal of labor is to have a child, and the goal of ripening fruit is to have ripe fruit. Okay, so it doesn't stop until that is achieved. Negative feedback, on the other hand, is the opposite. So again, if we're looking at this line representing homeostasis, and we come to this example of blood sugar, for most of us, sometimes our blood sugar is low. We eat something. Sometimes it's high. Sometimes it gets a little higher. Sometimes it gets a little lower if we don't. But we're always sort of coming back to this line. Okay? But in certain situations, if you eat a cookie and you're not, really a carb eater, I guess, your blood sugar is going to spike. Okay? So it drifts away from homeostasis. But there are negative feedback signaling molecules and pathways that can bring us back to homeostasis. So an excellent example of this is the dual effects of insulin and glucagon. So insulin basically does this. It brings your blood sugar down by signaling for your cells to take up sugar. And the opposite of that is glucagon, is if your blood sugar gets very low, glucagon can stimulate the production of blood sugars so that you're drifting back towards homeostasis. Okay, so that's an example of negative feedback. Negative feedback always brings you back to homeostasis. Okay, so again, a quick reminder. Let's emphasize the big ideas here. Feedback is a very important biological idea. It's in a lot of things that we do. It's not necessarily important to, study the physiology of insulin and glucagon. Most likely the College Board will give you that preview, and your job is to think about biologically how homeostasis is maintained. Okay, so in closing, cell communication is biology. For me, from my class, this is the end goal, is to get to cell communication so that we can talk about this. Cell communication, signal transduction, it covers every aspect of AP biology. Every single part of the pink book can be discussed from a cell signaling standpoint. And yes, that even includes ecology. Now, we're not going to have ecology on this year's test, but organisms and how they interact, a lot of that is based on cell signaling pathways in their cells and how they themselves as organisms are able to communicate from there. So obviously we don't have time to get into that. This video is already rather long, but yeah, I hope this helps. And if you're not necessarily from my class, I hope it was helpful. And for my students, thinking of you guys, I hope everything is well. Okay, talk to you soon.